Hello everyone, how are you doing? I hope everyone is doing good. So let's begin with another video of inheritance and variation. So in this video, we are going to cover incomplete dominance and codominance. In the previous videos, we had covered what is genetics, then we discussed about Gregor Mendel, who is the father of genetics, and we discussed his work regarding genetics, what experiments he did, why he chose the pea plant, okay, the 7 contrasting traits of the pea plant, and then we discussed about the certain terminologies. We saw that certain terminologies which are very important in the genetics chapter. Then we learned about monohybrid cross and dihybrid cross and the laws that were derived by Mendel through these crosses. That is on the basis of what observation and what inference he drawn from, got from those crosses. So law of dominance, law of segregation, and law of independent assortment. You should be knowing these 3 laws. If you have still not watched the video, please check those videos out. Because now in this video, we will begin with deviations from the Mendelian genetics, that is incomplete dominance and codominance. Okay, incomplete dominance and codominance. What will happen in this 2 example is that we are going to see examples wherein these will not follow. There are certain organisms, in fact, many organisms which do not follow Mendel's law of inheritance. Okay, so the first law that we saw was dominance, right? So if capital T and small t is present in a hybrid condition, we know that the capital T will be expressed, right? Because this is the dominant allele. But there are further studies were carried out after Mendel put forth these law of inheritance. So scientists came up with other examples in the universe, plants or animals, which do not follow this law of dominance. Okay, then another law was that gametes segregate, okay, before intermixing. So that also was not followed by some of the organisms. Yes, so that is what we are going to see in this video. Okay, deviations from the Mendel's law. Okay, so in that there are many examples. In this video we will cover 2 very important for understanding, and with respect to exam point of view also, you are asked difference between the 2, or you are asked give representation of incomplete dominance or codominance. Okay. Let's begin with the first example, that is incomplete dominance. What happens? See, first we will see the name. Dominance is that now we are aware that the dominant allele of the 2 pairs of the genes will be expressed. Incomplete means what? Only partial expression of that dominant allele will be there. Okay, only partial expression of that allele. So it is the inheritance in which the offspring shows characteristics which are in between, okay, characteristics which are intermediate between the 2 extreme parental combination. Okay, so if let's say the plant, one parent is homozygous tall and the another parent is let's say having 2 small t, then the hybrid condition will have this type of genotype, right? One capital T and one small t in F1 generation. That is how we know from our crosses, previous crosses. What will be the phenotype? If it is following Mendel's law, tall. But in incomplete dominance, the plant will neither be tall, neither be dwarf. It will be somewhere in between. Okay, so that is what we mean by incomplete dominance. Fine. So it is the inheritance in which the offspring shows characteristics which is in between, intermediate between the 2 extreme parental characteristics. Okay. We will see the example, the cross of in one of the examples of 4 o'clock plant. You have 2-3 examples over here. 4 o'clock plant color, flower color in the snapdragon plant. Another name is dog flower or anti species. You need to know the example thoroughly. One of the most commonly asked one line or MCQ in exams. Okay. This is the plant example which shows incomplete dominance, and another example is Mirabilis jalapa. Another name for Mirabilis jalapa is 4 o'clock plant. Now what happens in this? Let's just see the cross. You have to know the cross. You have to practice the cross. It is asked in exam. Okay. So let's begin. Let's take two parent. Don't look at the whole screen. Just focus on step by step what I am saying. So first we'll select the parents. Okay. Now the parent one, we will take the red dominant. Okay, having red color. The another we will take white color flowers, which is recessive. Okay, it is having two small r's, so homozygous, recessive, one parent, and this is homozygous dominant, showing giving red color to the flower. Now, when they are crossed, so first the gametes will segregate. So either you can write two separate RR, capital R, and two small r separately, or here just for representation, one is enough. It is understood that how many alleles are there, total 2. So 2 alleles will be there. And in F1 generation, possibly how many are formed? Total of 4 here. So this R will combine with this R, you get capital R, small r, hybrid condition. Similarly, when this R combines with this R, you get the same thing. When this R combines with this R, same thing. When this R combines with this R, same thing. Okay, so we just represent one here for understanding. Fine. So now, what do you see in the first generation? In first generation, the genotype is normal that we have been studying in monohybrid and dihybrid. Obviously, it will be capital R and small r. But what was observed by looking the phenotype was that the flower was showing neither complete red color, neither complete white color. It was somewhere in between. It was pink in color. It was what? Pink in color. Okay. So all the flowers that were, produced in F1 generation in the Mirabilis jalapa, all the phenotype of F1 generation were pink. This shows that it is not following Mendel's law. If it was following Mendel's law, then all the flowers should be what? Red in color, because R is the dominant allele. So this is an example of incomplete dominance. That is, the dominant allele R is not capable of expressing itself to the fullest. That is why small r will be also expressed. It will express some of it white also, and that is how the color of the flower is pink. Okay, nor red nor complete white. And then when we carry out the F2 generation, same like monohybrid cross or dihybrid cross, since here we are dealing with only one character, so you will get the result more or less like the monohybrid cross, the genotype ratio. There will be a difference in the phenotypic ratio. Now you take the same flower, two flower from the F1 generation. One you will consider as the male, another you will consider as the female. So this is how you will represent the gametes separately. You will put it into a checkerboard. So let's say here you represent the male, and in this vertically you represent the female. Okay. So capital R, small r. Yes. Capital R, small r. So when you cross capital R into capital R, you get two capital R. Then this into this will give you this. Then this into this will give you this. And then this into this will give you two small r. I will just erase everything so it becomes clear. Okay. So this is what you get in. This is what you get in your F2 generation. Okay. In F2 generation, you get the parental combination also. The parental combination are also seen, and hybrid conditions are also seen. So pink will also be there. If two capital R, definitely red color. If two small r, definitely what is the color of the flower going to be? White. And if hybrid condition, they will neither be red, okay, they will nor white. Obviously, white is a recessive character, but R is capital R in presence of the recessive allele is not able to express itself completely. That is why the name incomplete dominance. So pink. So what will be the genotypic and phenotypic ratio? So phenotypic ratio, 1 red flower, 2 pink flowers, and 1 white flower. So 1:2:1 phenotypic ratio, and genotypic ratio also will be the same, right? 1:1. Okay. So phenotypic and genotypic ratio in case of incomplete dominance will be the same. So this is example of incomplete dominance, Mirabilis jalapa 4 o'clock plant or snapdragon plant. They do not follow Mendel's law of dominance. They show incomplete dominance. Moving to another example of deviations from Mendelian genetics is the color of the cattle. Okay, another example is also human blood grouping. What happens in co-dominance? Now you can relate the term. In the earlier example, it was incomplete. Now it is co. Co means what? Equal expression. So in which inheritance in which both the alleles of a gene are expressed in a hybrid. Okay, both are expressed. There, the capital R was not capable to express completely. That is why instead of getting red color, lighter version of reddish pink was observed. Here, both will express themselves equally. Their partial expression was there of the dominant allele. So cattle, and even if in our blood grouping system, you have A blood group people, B blood group people, O blood group people, AB blood group people, this also shows co-dominance. We will take the example which is there in your syllabus, that is the cattle cross, the color of the cattle cross. Sometimes you can take black. Instead of black, you can have brown also. So that depends. Okay, usually it is brown. So we can take 2 capital B. Here the letter represent is 2 capital B. In your textbook, it can be round when we are considering brown color. So 2 capital R is represented. Okay. So round means a slight brownish, blackish, brownish color. Okay. Blackish, brownish color. Blackish, brownish color. Fine. So we can just take this example. No harm in that. So let's take the parent combination first. So in parent combination, 1 cattle is black color, or you can say brown color, 2 capital R or 2 capital B, whatever you want to take the example, and white obviously will be the white. Okay, when you cross, first you will segregate the gametes. So this is how you will represent 1 capital B, 1 capital W. What do you see over here? What's the difference? Both of them are dominant for their trait. White is also dominant because 2 capital W, no 2 small w. Capital W we have written over here. So 1 capital B, 1 capital W. What do you get? All spotted. You might have seen cattle. There are certain cattle which have patches on them. Black and brown, black and white. They have patches. White patches are also there. Brown patches are also there. Or black patches are also there in place of brown. Okay. So that is a hybrid condition. White color is also expressed and black color is also expressed. So you will get all spotted, or you can just say it round, RAON. Sorry, there is a problem with my cursor today. That is, I'm not able to use it properly. So F1 generation, you will get all mixed. The patches all spotted. Now you take the same B capital W and capital B capital W in F2 generation. Now you do a cross. This cross is represented a bit tilted, but that's the same checkerboard cross. That is the 4 square that we made. Okay, we make. So you do the multiplication, capital B into capital B into capital W, then this into this, and then this into this. These are the 4 possible progeny that you will see in your F2 generation. So you will see the parental combinations also, and you will see the hybrid conditions also. Okay. parental combination black and white, and then patched one or spotted ones. Okay. So here also the phenotypic and genotypic ratio will be the same. 1:2:1. One black, two spotted, and one white. Similarly, genotypic ratio also is 1:2:1. Okay, so in incomplete dominance and codominance, the phenotypic genotypic ratio is the same. Okay, only difference is that there the dominant color was unable to express, the dominant trait was unable to express itself completely. That is why some intermediate color was observed in the F1 generation and also in the F2 generation in the ratio of 2, right? Codominance, both the alleles are dominant individually, and when you cross those individual type of cattle, what do you see in F1 generation? You will see both of the traits expressed in the F1 generation. So white patches also and brown patches also. So this is the example which is there in your textbook. This is the with the black cow, and this is with the brown cow. So I've included the both. You may take whatever example you feel comfortable like. Okay. Sorry, this has to be the capital W. Capital R and capital W are present together, so they will express themselves equally. This is due to codominance. Okay, it's like co is like both of them are equally participating. Fine, both of the alleles. So R, W, and this is how the cross will be done. You can call it as red or brown, or whatever you may. It's like a reddish brownish color. So this is about codominance. So that's all for this video. Before closing the video, we'll have a look at the difference between the incomplete dominance. Again, one of the most importantly asked questions. So we will just go through the difference between. What happens in incomplete dominance? Both the genes of the pair, okay. So there it was red, capital R, and small r, white. Both of them express partially incomplete, not complete, partially half in F1 hybrid. That is why the color is neither red, neither white. It is somewhere white in between. Sorry, pink in between. What happens in codominance? Both the genes of the allelomorphic pair, that is, let's say, B capital W, express themselves equally. So this is the keyword that you need to know between the difference. Partially, incomplete, and your equally. The phenotypic typic effect of one allele is more prominent than the other. Though partial expression is there, but it is more towards the reddish, pinkish is more towards the reddish. That is why one allele is more prominent than the other. Here, both effect of both the alleles is equally prominent. So you will see white patches in equal, and you will see red, brown, or black patches in equal. Here, blending or intermixing of the two alleles can be observed. That is why you see pink colors. Some white mixed with some red. The genes blending or the two intermixing of the alleles can be observed. That is how you get pink colors. Here you will see no blending. You see different patches of white, independent patches, and of white, independent patches of red. So intermixing or blending effect of the two alleles is not observed. So that is how you see the spotted one or the round color in the capital. Okay, so 2 marks ke liye this difference can come. Very important. You need to learn this. So that's all for this video. In the next video, we will see another examples of deviations from Mendelian genetics. So these were the examples which will not follow Mendel's law of dominance. Okay, another examples in the topic of pleiotropy and multiple allelism we will see in the next video. So thank you all. Take care. See you all in the next video. Bye.